some highlights as it relates to, I have no idea why this is doing this. It's really annoying me. Okay, so unknown reports. So on the first page, some people did not accurately describe or draw their negative stain. Remember, for a negative stain, what should be colored? The background. The background should be colored and your cells should have been clear. You needed to not only draw this, but also write this, right, for your negative stain. So some of you guys may have lost some credit on that. The other place where people, and even though I told most of you guys, people sometimes lose points, is for the capsule stain. It says, right, draw a picture of capsule stain, label cell and capsule if present, right? And again, your description should have had that your background was dark, right? Your cell was stained. The capsule was not stained, and that it surrounds your cell. Remember, you're going to see slides like this again, and you need to be able to make these identifications on the final exam, right? I'm going to have a capsule stain, right? Or maybe even another stain, and you got to tell me which one's the capsule stain and which one has a capsule. Right? All this stuff you did for your organism, you're going to do again on the practical exam, right? You're going to look at slides again. You're going to determine, does it have a capsule? Is this gram positive? Is this gram negative? Is this acid fast or non-acid fast? Any questions on this page of your guys' report? Okay. So I didn't update my date on this, but you know, it holds true, unfortunately, semester to semester. I don't know how to fix this problem, y'all. Right? I don't know why you guys don't label stuff on this report. It's like the same things I'm saying over and over again. The genus and species names of organisms should always be italicized or underlined, which is kind of hard on your guys' report because there was a line for you guys to write. But I just want to emphasize that that's important, right? How we write names of organisms, right? That you should always underline it when you're handwriting the full name of an organism. So I'm not really going to ding you guys. I didn't ding anybody on that, on their report. But I definitely want to emphasize that as you move forward in your careers. And I type a lot, so, you know, I get the pleasure of being able to italicize very easily, right? And I have to admit that a lot of times when I write, I don't underline it, and I really should. So the word bacillus, for example, not only refers to a genus, right, but it also refers to a shape that we commonly just refer to as a rod, because again, it's kind of confusing when you're talking, right? Well, what are they talking about? Are they talking about the genus, or are they referring to just the shape of the organism? The good news with bacillus, that genus, they're all rod-shaped. That's how they got that name, right? And some of you guys had a bacillus. And then others had cocci, right, that are spherical in shape. And then some even had arrangements, right, where they were in chains or in clusters, right, and you needed to be able to identify that, know that terminology. So the same thing on the practical exam, right, you're going to have a slide where you're going to see a morphology, right, and you're going to see an arrangement, and I'm going to ask you what's the term that we use for what you're seeing, right? Is it staphylococci? Is it streptococci? Right? Is it diplococci? Is it streptobacillus? Right? What are you seeing? Right? So make sure you review that terminology, right, as it relates to. And of course, these are the only choices we had in this lab was either cocci or a rod, right? We don't have any of the other shapes, but you should remember those and review those as well, right? So gram, by the way, too, should always be capitalized when we're talking about the gram stain. Lowercase refers to the measuring of units like a gram, right, that weight that's used. So again, doesn't go away, gram stain, as I tell my lecture students, I'm going to be talking about it all semester long, and this is the one time you'll see Miss Erica cry, is if you don't still understand this concept, right? So our gram-positive, remember, really thick peptidoglycan layer, and they're going to stain what color in the gram stain? Gram-positive will be what color? Purple, right? They're going to retain the crystal violet iodine when we decolorize, right? Unlike gram-negatives, right? They come out what color? Pink, because remember they decolorize in the decolorization step, and this is due to the problem of not having a lot of peptidoglycan, right? And what happens to this extra layer, and what do we call this extra layer? That's the outer membrane, right? And it gets dissolved by the ethanol, right? And so the crystal violet iodine is not trapped in, it washes out. Then we counterstain them with safranin and they appear pink. So this is still important information. Although routinely performed in clinical labs, this technique is sensitive and therefore requires both positive and negative controls on the same slide, right? So you have to perform it correctly, right? And you guys are among a group that you're lucky, probably the last group that's not going to have to do a gram stain on the practical exam. But future students are going to be doing a gram stain. It's that important. So in addition, our selective media helped us out, right, with confirming our gram stains, because again, they can be kind of tricky even with controls sometimes, right? I think some of you guys had some difficult results to read. So again, review all your stains, right? Gram positive or purple, gram negative or pink. What does a capsule stain look like? What does a negative stain look like, right? What do endospores look like in the Schaefer-Fulton method that we use? What does acid-fast versus non-acid-fast look like, right, in an acid-fast stain using the Zealand-Neathan method that we use here at Delgado? And again, arrangement. Notice this is Diplobacillus, 2 rods right next to each other in pairs. This would be what? What are all these guys? So these are rod-shaped, right? That would be Streptobacillus. Yeah. This wouldn't be an arrangement. They seem to be all by themselves. They're not staying next to each other. And when you're trying to figure out arrangement, what you would do is you'd scan around on your slide and you would look for the most complicated arrangement, right, that repeats itself. Very easy to see here, right? This one not so easy, but definitely some clustering there, right, with those cocci. So you might be looking at staphylococci. And this one, you're definitely staphylococci for the little blue ones. So most bacteria that we work with are encapsulated, that we worked with here at DCC. Few of them, right, have spores, just our bacillus. And then we actually only had one mycobacterium, right? Again, I want to change that in the future. So organisms that are motile, right, that can self-propel, generally it's by means of one or more flagella. So that's one of the things that you guys can look up, right? You guys know if your organism is motile or not. You know what your organism is, right? For a little fun, right? Do a little quick Google search, right, for your organism and see what you can find out. How many flagella does it have if it's motile, right? How are they arranged? And remember, that was some terminology that we learned that you're responsible for. So, remember for this test, if it stays where it is, it's negative, right? If it's spread out 360 degrees, you turned your tube, right? You didn't have a bad stab or anything, that it was motile. Some of you guys had to do additional testing because your organism didn't really perform well with that media, and we looked at it under the microscope. Right, so we could confirm that way. So when drawing the FTM tube, sometimes people do, again, don't label. You've got to label, right? You've got to label, right? You should have labeled your oxygen zone at the top and your non-oxygen zone at the bottom of your tube, right? I think I might try and add some these little bar things next time. I don't know. I don't know what to do. I even did drawings on the board, I think. People don't label. Right? Very important, right? Good note-taking skills. Very important. So for here, for us, again, we did this to ask the question, can your organism grow anaerobically? And remember, we really don't have strict anaerobes here at Delgado or microaerophiles. Too difficult to maintain the environment to grow them in. Right? So we had obligate aerobes, right, where they only grew at the top in the oxygen-only layer. So we know they're only using oxygen for their metabolism. We had a whole bunch of facultative anaerobes, and if you were lucky, you saw a greater amount of growth in the oxygen zone because it produces more energy, right, via aerobic respiration. But it can ferment, right, without oxygen. So in this non-oxygen zone, you also see growth. So this guy can grow anaerobically, hence its name, facultative anaerobe. If you didn't see this distinction, there was another test that helped us out in knowing if your organism was a facultative anaerobe versus an aerotolerant anaerobe. And what's different between these guys? So what do you see different in these tubes? The growth pattern's different, right? So just one. Go ahead, Cynthia. What were you going to say? The aerotolerant, the growth is even throughout the tube. Do you have anything else to add, Elizabeth? No? Okay. So why is that, though? What do we know about these organisms as it relates to oxygen versus no oxygen? Why is it uniform? All right, hold on. One person. I'm going to give Grace a turn. Go ahead. It can grow in both the oxygen and non-oxygen zone, as we see, because growth is uniform throughout. But why is it uniform as opposed to this guy? He has more growth in the oxygen and as opposed to the non-oxygen zone. Hold on. Naomi, your turn. So this guy prefers oxygen? What about the aerotolerant anaerobe? It's not affected, which means that it, unlike the anaerobes, we're not going to kill it off, right? But is it using oxygen? No. Right? Because if it was, you would see an increase in growth in the oxygen zone. These guys are usually fermenters. Right? So they're doing the same in the oxygen zone and the non-oxygen zone. They're doing fermentation. They're not doing aerobic respiration at all. So what other test told us if your organism had an electron transport chain and could utilize aerobic respiration? What other tests did we do? Which test is related to the FTM test? Nope. Catalase. Because catalase is what? An enzyme. Should be my next one. No, crap. That's okay. It's an enzyme. I'll leave it on this page. It's an enzyme, right, that aerobes have, or organisms that can do aerobic respiration like facultative anaerobes. Because what does it do? Here, I'll skip ahead to that slide because I know I've got it. Here's catalase. Catalase does what? Ooh, test is next week, y'all. Alright, Jessica's turn.
It needs catalase to use aerobic respiration.
Why does it need catalase? So what does catalase actually do? I don't know. Alerica?
Catalyzes the of water and oxygen with hydrogen peroxide.
Yes. So it breaks down hydrogen peroxide, which is a damaging form of oxygen, right, into water and safe gaseous oxygen. That's why if your organism was positive, it did what? It bubbled. It bubbled, right? And that told us that your organism has an electron transport chain. It can utilize oxygen as its final electron acceptor. So that electron transport chain, sometimes the electrons hop off before they're supposed to, and they form these toxic forms like hydrogen peroxide. There's one other one, enzyme, that we didn't test for that helps protect against toxic forms of oxygen. For superoxide radicals, it actually turns superoxide radicals into hydrogen peroxide, which then can be broken down by catalase. What's the name of that enzyme? Yes. Superoxide dismutase, right? We didn't test for that, right? But in addition, these organisms that have catalase will have that enzyme as well. So aerotolerant anaerobes, right, can tolerate oxygen, but they're not utilizing it. So the person that had that organism that was catalase negative had the aerotolerant anaerobe, right? Because they don't need catalase, because they're not generating a lot of hydrogen peroxide because they don't have an electron transport chain that's using oxygen as the final electron acceptor. So they're not generating a lot, but they do have superoxide demutase, because that's going to protect them from that toxic form of oxygen that could potentially be exposed to. And that's why they can tolerate oxygen, but because they're not using oxygen, they don't have to have catalase. Does that make sense to you guys? If it really doesn't, please let me know, because test is next week. Okay, so let me back to the next set of tests. So phenol red broth. Typically we do this to figure out if organisms are doing what with glucose. Catalyzing it how? So fermentation. A lot of times we're looking for fermentation. What are common byproducts of fermentation of sugars? Alcohols, acids, and gases. What can we detect with the phenol red test? Gas by the derm tube, right? Trapping the gas and displacing the media and forming bubbles. And acid, because what is phenol red? pH indicator. And what color does that pH indicator turn in the presence of acids? Yellow. Is that the only thing we can tell in this test? We can tell if it goes the other way as far as pH, if it becomes basic, right? If it's that magenta pink color, that tells us they didn't ferment the sugar. Instead, they did what? They deaminated the protein, which produces what compound that's basic or alkaline? Ammonia. And therefore, again, pH indicator in alkaline or basic levels, we know that that happened because it turned pink. Okay, the citrate test. What are we testing for? What does your organism have if it's positive for this test? Embedded in its membrane. Citrate permease. Right, so this is a protein embedded in its membrane. It's a critical enzyme needed for being able to utilize citrate. Right, it helps citrate diffuse into the cell. The media, when the organism utilizes it, becomes basic, alkaline. When ammonium is consumed from the media, right, and this causes the basic reaction, very similar to what we see in the phenol red, but the problem is we're using a different pH indicator here, right? We're using bromenthal blue. So our tubes go from green to blue if they're positive. What happened to your organism if it didn't have citrate permease? The tube stayed green because, again, it didn't utilize the ammonia. But why didn't it utilize the ammonia? It died because, remember, we were very evil with this media. There was only one carbon source to choose from and one nitrogen source. So if they could not utilize that carbon, they died, and therefore also did not utilize the nitrogen source. Dead bacteria don't grow, right? They're not going to break down the ammonium, they're not going to produce the alkaline product. Make sense? So some of you guys, remember I told you, if it was a green tube and you thought you were seeing growth, was that really growth on your slant? No, you probably put too much bacteria on your slant to begin with. You probably have done what we call a heavy inoculum. With this test, some scientists will not even use a loop. They'll use a needle to make sure they pick up just a tiny bit of bacteria to put onto the slants. I wasn't so concerned about this for you guys, because usually we have the opposite problem. Not enough bacteria. Okay, so we already talked about catalase, so we'll skip over that one. I might want to change the order on that one next time. So, methyl red test, right? So we use the MRVP media, which contain glucose. For both those tests, remember our positive result was what color? Red. But they mean different things, right? Phenol red is a pH indicator. We did a pH test. And for this, if it stayed red, the pH is 4.4 or lower. That's very acidic, which means potentially you've got a lot of acid in that tube, right, from the fermentation of the glucose. We refer to these organisms as mixed acid fermenters, right? That's the test we're doing. Are they a mixed acid fermenter if their methyl red test is red? So methyl red positive organisms should be acid producers, which means in our phenol red test, they should have been acid no gas or acid gas, right? But the reverse statement is not true. Phenol red positive does not necessarily mean they're going to be methyl red positive. Why is that? Anyone know? So, two different tests, they both have pH indications in them, right? But is the pH range the same? No. So in comparison, right, methyl red, as we said, it's got to be 4.4 or lower to be positive. What does it have to be to produce acid to make it yellow for phenol red? Anyone know? .8. That's not very acidic, is it? That's just barely acidic, y'all. Neutral is what? 7. So for the phenol red test, they just got to produce a little bit of acid and that tube's going to turn yellow. But is that enough acid that when we do the methyl red test that it's going to be positive? Sometimes no, right? Because you've got to be 4.4 or lower for methyl red to stay red. Right. But definitely, if you're methyl red positive, you should be phenol red positive, right? Because that's a much higher acidic pH than 4.4. So these were a couple of questions on your unknown report. That I didn't take off credit if you got them wrong, but I wrote in the correct answers. And this, again, it was really get you guys to get to that higher level of thinking, of comparing and contrasting things and really understanding how these tests work. Anyone still confused on that? So, the Voges-Proskauer test, we're testing for the ability of your organism to do what type of fermentation? What are they doing? It's not mixed acid. They're breaking down glucose into what product at the end? 2,3-butanol. How do we know your organism has this ability to make 2,3-butanol? And again, this is the only test that we do for fermentation that produces a what? What is butanol? That's an alcohol, right? It's the only test we do to determine an alcohol fermenter. But do we actually test to see if 2,3-butanol is present? No, we test for its intermediate. What's its intermediate? Acetonin. So the reagents that we add, the alpha-nithaphthonol, the KOH, right, potassium hydroxide, and oxygen that we add by flicking our tubes, combines with the diacetyl and the guanine to produce the red compound that gives us a positive test. But this is a chemical reaction that needs to happen, right? So this is why we waited 60 minutes to make sure that it had enough time to happen to produce enough color that we could see it with our eyes, right? There could be a sensitivity issue here if we didn't wait long enough, where we don't see the red, but it's actually there. Just not in a high enough concentration that we see it with our eyes. So our selective media, right? Definitely review your quizzes, right? Some of you guys struggled on that last one. So we used antibiotics as a selective agent, right? It inhibited the growth of what? Did colistin and nelidiscic acid inhibit the growth of what group of organisms? No. The antibiotic plate. It was antibiotics with the blood, the whole red blood cells. Colistin and nelidiscic acid would kill off gram negatives. So therefore, only gram positives will grow on that plate, right? And this is important, right? If we're selecting, so if the media contains a selective agent, we're first selecting for a particular group. But they also have a differential component that distinguishes the survivors, right? So if your organism was gram negative, and in this case, I didn't even give you a blood agar plate, right? Because was it going to grow? No. So since it wasn't going to grow, would you be able to determine whether it was hemolytic? No. You'll notice on your lab report it says cannot be determined because no growth. Right? And that's what you need to say about your organisms that didn't grow on these selective medias, right? Is you don't know. You can't say it's not hemolytic. You can't say it's not a fermenter. You don't know. We could not test for that. Dead organisms don't do these things. And I got news for you. Some of you guys that have gram negatives, they are hemolytic. But we can't determine that because the plates we use select only for gram positives. If we remove that selective pressure, then we could ask those questions, right? So for instance, for the mannitol plates, what was the selective agent? Why did only a particular group of organisms grow on the mannitol salt plates? What in that plate was inhibitory? Go ahead, Grace. Sodium chloride. But this, you guys did get dinged on your report. Because you can't just say salt. Most of the media we work with, guys, has salt in it. High salt concentration. That's what the up arrow means, by the way, on your lab reports. I use shorthand. If you have that on your report. High concentration of salt. If we remove that high concentration of salt, all the organisms would grow, right? Then we could ask the question, is yours a mannitol fermenter? But again, we can't ask that question for most of your guys' organisms because what happened to it in the presence of high salt? It died. Right? So mannitol salt, right? We're selecting for salt-loving, salt-tolerant organisms because of the high salt concentration. And then amongst those survivors, right? If yours is salt-tolerant, then we can ask the additional question, does it ferment the sugar mannitol? If it does, we know a product of sugar fermentation is acid, right? What pH indicator is in these plates? Well, it turned yellow. We've dealt with that before. For acid, yellow, that's which pH indicator? It's phenol red. And remember, I posted an announcement, and I highly suggest you guys look back at it, how detailed your description should have been for your results on your organisms, right? You really need to understand every aspect of these tests and how it's telling you what it's telling you. So I had wrote out for mannitol salt all the different possibilities and an announcement for you guys to go back on Canvas and look at that. Right? Really helps step through. And for every single test, you should be able to write and understand in that level of detail.
I don't know why you took off on here.
Because you didn't include everything in your description that you needed to. So do you guys want me to go to that? I'll actually go to that now. I think it's a good thing to go to.
Because I said, like, it can't tolerate salt, so it's killed, and it doesn't generate acid in parts because it did turn yellow.
Right, but that's not the reason. Did yours grow on those plates?
No.
No, then you could not make that distinction. You should have said it could not be determined because of no growth. Yeah. Okay, so let's see. Announcements. So, which brings me to a very important thing that I need you guys to do. So here's unknown reminder about unknown lab reports is where this write-up is that I did. Right? So, is that small? It's kind of small. Let me make it bigger. Okay. All right, so here's the three possibilities we could have had. Right? You had no growth. Right? Color of the media, therefore, we cannot determine, that's not applicable, right? If you have no growth, of course you're going to get no color change. So if you had no growth, it means that your organism is salt intolerant, right? And you cannot determine result 2, fermentation, because no growth happened. So your explanation would be my organism is salt intolerant because of the high level of salt in the media killed the bacteria, resulting in no growth. Right? So we're explaining the salt, we're explaining why we got the result we got.
Organism cannot tolerate high concentrations of salt.
Yeah, so you got that. So you didn't lose points on that.
Yeah, it was 0.
Right, this next part, right? So mannitol fermentation cannot be determined because the bacteria died. Right? When sugars like mannitol are fermented, acids are produced and can be detected by a pH indicator. The pH indicator, phenol red, would turn yellow in the presence of acid. So you have that information, you're just missing the key thing that you really could not determine that for yours. Does that make sense to you guys? Right? So for another example, what could have happened is you could have had a lot of growth, but no color change from control. Right? The media didn't change color, which was what's supposed to have happened for Elizabeth. Right? But she did a really good job of writing out, right, and explaining what I had told her is that, hers went too long. So if that's the case, my organism is salt tolerant, right? If it grows, it's salt tolerant because it grew in the presence of a high concentration of salt. This bacteria, on the other hand, doesn't ferment mannitol because there was no color change of the media. So again, we repeat, how do we know that what no color change means? We know when sugars are fermented, acids are produced, the pH indicator would turn yellow. And we can even go one step further here, right? The media did not turn yellow, therefore it didn't ferment mannitol, right? My organism is Staphylococcus epidermidis, confirmed by other tests completed in this report. Right? This one would really help someone that had Staph epi really narrow it down to that organism. Or if they had Staph aureus, or one of our bacilli that will also grow on these plates and sometimes turn it yellow, you again would have a lot of growth, but in this case you had yellow, right? Your plate was yellow, which tells you, again, it's salt tolerant, right? And the pH change tells you that acid was produced, so therefore sugar was fermented, right? And then that could have confirmed that yours was Staph aureus or that your organism is a bacillus. one of the 3 species. Do you guys see how every single detail of that test is explained out? Okay, so back to here. So that's mannitol salt. Let me go back to my slideshow, current slide. So EMB, right? Remember, the formula that we used in lab only contained lactose, not sucrose. So we were asking lactose fermentation here. So again, this is a selective media. What makes this media selective? What's in it that kills off the other group of organisms? Right, the EMB stands for eosin methylene blue. Those 2 dyes are toxic to who? Gram positives. It's toxic to gram positives, so who grows on these plates? Gram negative. So if you had a gram negative, it should have been confirmed by the fact that it'll grow on this plate. So if you had a gram positive, did it grow on this plate? It shouldn't have. So can you ask the question, is it a lactose fermenter? No. No growth. You stop there, right? The rest cannot be determined. If you do have a gram negative, then we can ask the question, is it a fermenter? And actually we can ask, how vigorous of a fermenter is it? Who wins the prize on fermentation of lactose? Who is the big guy who ferments like crazy? Lactose. It's this pretty one right here. Who ends up being metallic green? E. coli is one of the ones that is a really strong fermenter, right? So you see that metallic green sheen, right? Might be black or very dark purple. That's an indication that it may be E. coli. What does it mean that if there's no color change, right? No of the pink or the purple that we would see, or this metallic green, right? What if it kind of blends in or clear with the plate? Is it fermenting lactose? No. If it's pink or purple like you see here, is it fermenting lactose? Yes, it's what we call a moderate fermenter, right? It's not a vigorous fermenter like E. coli that's going to go to the metallic green, right? It's a moderate fermenter. And it's the dyes that interact with the acids that produce these color changes that allow us to detect levels of fermentation of lactose. So again, the sheep red blood cell has CNA in it, which is calypsin and nalidixic acid, 2 antibiotics that select for gram positives grow, gram negatives are killed. So again, if you had a gram negative, all you know and all you confirmed is that it's gram negative because it was killed by the antibiotics. You cannot determine hemolysis. Now, if it was a gram positive, then you can determine hemolysis. Remember, there's 2 types of hemolysis. There's alpha and beta. What does beta mean? Bad. More detailed. It's a good start, though, right? Bad. What do we mean by bad? What did it do to the red blood cells? It completely destroyed them, right? Annihilated them. So beta, you see that complete clearing. Alpha is incomplete. You see that greenish discolorization to the red blood cells, right? Which means it just poked holes in the membrane. What is gamma? Yeah, but it wasn't inhibited by the antibiotics, right? It would have grown. The organism is gram-positive, but gamma means that it's non-hemolytic, right? It didn't destroy the red blood cells. So your question in your report actually was really simple, was, is gamma hemolysis, does it lyse red blood cells? The answer is very simply, no. Typically, we use this for staphylococci and streptococci and enterococci, right? to help differentiate them. Justification of inconsistent results is important, right? And as important as your correct identification. So that's your very last page. Right? It's important as a scientist to keep good notes, right? Make notes of things as you go. So let's do a little bit of practice next. So for the final exam, you guys have this sheet. I handed it out, right? Reread the syllabus to refresh your memory as to what the instructors expect you to learn for each exercise. Right? Syllabus isn't just something to just put in your binder and not ever look at. We put a lot of time and effort into that. As always, your homeworks, your quizzes are excellent study guides. You should know all terms again, right, and be able to spell them correctly. Your graded unknown report is an excellent study tool. In PowerPoint lectures, each slide that's entitled Scientific Method, right, is a really good overview of that exercise and a really good, slide to key in on when you're going through your study process, and see if you can go through those steps, right? Like, it's animated, so see if I remember, why did I do this? How did I do this? What were the results, and what do these results mean? Create your own tables of information, right? And summarize the essence of each exercise. Right? So as we said, why did you do it? How did you do it? What does this mean? Exam covers all exercises after midterm. Most experiments helped you, right, identify your unknown, but some were standalone exercises that were not associated with your unknown identification. PGLO, what we just went over this morning. ELISA, what we're going to do today. The antibiotic sensitivity test, right? We didn't do that on your unknown. Although that would be kind of cool, huh? Maybe we'll might make some changes. The dilution exercise we did, right? So these are all written out too for you, right, as a reminder. So these are all the ones we did on your unknown, right? 12 different tests. And then the standalones is the standard plate count, as I just said, the Kirby-Bauer antibiotic sensitivity method we use, bacterial transformation, which we did last week, and then ELISA, what we're going to do today. You should understand the process of identifying an unknown organism. All the steps you perform to identify your unknown will be on the exam, as I've said. You need to be able to evaluate stain results. Be able to classify based on shape, arrangement, and morphology on stained slides. Given a table of known data, so of course that table that's in your guys' unknown lab report, which you're going to need today to do today's activity. So make sure you take out your lab report. If you, oh no, it is actually on the back page. Okay. So that table I would give you on the exam. By no means do we expect you to memorize these results, right? You'll be given that page on the exam, and you'll do just as you're doing right now, right? Given information, figure out or narrow down to what organism do you think this is. So we're going to take a moment, and I want you guys to try and work out problem 1. So given these results and looking at your table of known data, if you have an organism that's gram-positive, phenol red, the code would be K, which means it ate the protein, right? And it's a pink tube. Motility test was negative, capsule stain was negative, and acid fast was positive. Should be a red flag going off right now. Nope. There's one test on this list that actually narrows it down to a single organism on our list. Tracy? Acid fast, right? Who's acid fast? One genus of bacteria, right? Mycobacteria. So on our list, it's Mycobacteria smegmatis, right? You could go through the process of elimination, right? But acid fast, if you look on our list, the only one, right, is Mycobacteria. So that one's quick and easy, right? What about the next one? This is going to take a little bit more deduction. So it's gram-positive. That narrows our possibilities in half, right? Phenol red is acid, no gas, so A-. Motility is negative. Capsule's positive. Catalase is positive. Citrate is negative. Who is that? Anyone got it yet? Ah. That only brought us down to 2, right? The 2 staffs. Pretty much the same on all those tests, right? Gram-positive, as I said, narrows it down into half of the organisms, right? Phenol red eliminates those 2. Citrate and motility narrow it down to these 3. And then catalase narrows it down to those 2 guys. So, oh boy, right? Mm-hmm, absolutely.
Some of the lab exercises, you ask, like, is it a sensitivity or a specificity issue?
Yes.
Or questions like that beyond that?
Absolutely. It's in your homework, right?
It is.
On your quizzes, right? The trick with that, you guys, sensitivity and specificity. If you're looking for a color change, it's usually a sensitivity issue, right?
But it was only not with more sensitivity issues than specificity.
Right. We deal with more sensitivity issues than we do specificity. Specificity means how specific is what you're testing for is. Is there a possibility that something else that could cause this reaction? So with the catalase test, is there something other than catalase that can break down hydrogen peroxide? There's a reason why we keep it in the bottle that we keep it in. What's unique about what bottle we keep hydrogen peroxide in?
Oh, so it was dark because of the light.
Yes. So light can cause that reaction, and metal, because remember when we performed that test, we put our bacteria on the slide first and then added the hydrogen peroxide. If we had added the hydrogen peroxide and put the bacteria with the loop, we don't know if it's the metal loop that's causing the bubbling or your organism. Those are examples of specificity issues. Right? And the way we perform that test, we can avoid one of those issues. All right, so we've narrowed it down to these 2 staffs. So Tracy and Elizabeth had these guys, right? So what helped you in narrowing it down? MSA, right? And what do we know? What do we know about MSA as it relates to staff? It's selective for those salt-tolerant staffs, okay. So both of them would grow, though, right? Both Staph epi and Staph aureus grow on mannitol salt plates. What's different about them as it relates to mannitol salt?
Aureus could grow.
Aureus will grow and do what? What does aureus do? What does aureus do, Tracy? It ferments mannitol, so your plate turned yellow. Elizabeth, what happened? What was supposed to happen with your plate? It was supposed to grow, but not turn yellow. Okay, so there's one more media that helped you guys out too to confirm that you had Staph aureus versus Staph epi. What other media did yours distinctly make a change to? CNA, right? So what do we know about one of these organisms as it relates to CNA? Tracy, it was yours. What did yours do?
It
Right. Staph aureus is beta hemolytic. It completely destroyed the red blood cells. That I don't necessarily want you guys to memorize, right? But, you know, it'd be a good thing to know about, right? But definitely, as far as it relates to the mannitol salt, you do need to know that. Right? But again, hers was beta, where no lysis happened for Elizabeth's. Okay, so let's try this next one. So, gram-negative, rods, acid, no gas, methyl red positive, capsule stain positive, citrate positive. What 2 organisms can you narrow this down to? I think, right? This is a narrow down one as well? Okay. I honestly, surprisingly or not, don't have these examples memorized.
Is it a ratio?
That's one of them.
No, actually, I guess this one is narrowed down to Serratia.
What about the other one? With the A.
It's not, I have a sentence.
No, it can't be. It can't be a bacillus because it's gram-negative, right? So right away we're negative, right? Phenol red brings us to these guys. Citrate eliminates them. It brings us to Klebsiella pneumoniae and Serratia. And the phenol red test tells us that it's Serratia. And actually, that one lately too, we've had good inoculations. It actually will start before the 48 hours. They'll actually start eating the protein. So that one's actually been a week K as well. So people who've had that one have known pretty much right away with their phenol red result. All right, so my last one must be a narrowed down to 2. Okay. Yeah, yeah. So yeah, you're right. It's kind of fun, right? I like it. Cynthia's like, no. So you guys want me to read the results? Does that help you? Or so gram-positive, phenol red, acid, no gas, rod-shaped cells, FTM test for anaerobic growth. It was negative, which means it only grew aerobically. Spore positive, so that narrows it down to the bacilli, right? And methyl red test negative. So our possibilities are 2 of the bacilli. Megatherium and Subtilis. So, again, spore brings us right down to those 3, the bacilli. Methyl red and anaerobic growth eliminates the Bacillus cereus and leaves us with Bacillus subtilis. So one test on here would help us narrow it down between these 2, right? So, yeah, the VP. Do you guys see that that's the one that's different? So one test from the chart, right, is the Voges-Proskauer test. But do we want to rely on just a single test? Right? We had some issues with some of those tests, right? So to help us out with those organisms, we do one more selective media to help confirm. And this I don't want you guys memorizing per se, right? I won't ask you one related to this. But if you look on your lab reports, right, as it relates, it tells you, right, who's beta hemolytic. And which one of those 2 is beta hemolytic? Yeah, subtilis actually. It's produced subtilis. So the people had Megatherium or subtilis, right? VP would let them know, but Mannitol, blood agar plates would confirm.
Can you go back?
Yep.
So magnesium was the one that's faded?
Subtilis. It's in your guys' lab report, but again, I'm not going to make you guys memorize that. Okay, so first part of the course, right? Whole bunch of staining, learning how to deal with bacteria, the darn eukaryotes that you don't have to see again, and you're glad about that, right? Lots of structural things, and remember, the second half is more physiology-like, right? What are your organisms doing? Lots of functional tests that we performed. And so remember, microscopy, resolution is important, right? Magnification is important. Contrast is important. This also is not on the final, though. We're not going to go back over and calculate resolution again, or magnification. And remember, we do bright field microscopy. So again, making tables, right, is a good way of organizing information, processing it, making sure you remember. So this is an example of a table that has all the stains, right? And why we use them, right? What results mean? What are the different steps? How do they work? Standard plate count. Right? Suppose to determine the concentration of cells, viable cells in a culture, the original culture cell density or concentration is referred to as its titer, right? And we do this in colony forming units, right? We count the colonies per milliliter. Remember, we want to stay within the countable range that we utilize for this lab, which is 30 to 300. We need to know the dilution factor of our plates and the volume that we plated. So you got to know this formula, right? You have to be able to calculate these things for the exam, right? So this is your key formula, right? Colony forming units, how many colonies you counted, the volume you plated, you divide that by, multiply by your dilution factor, right? How dilute is that sample that you took to put onto that plate that was countable, right? Make sure you guys have the old practice problems and all of that in recordings, right? So if this is tough for you, make sure you go back over this. So notice here, 0.1 into 9.9, that's a 100-fold dilution, right? And we just kept doing it. So, 100 times, and then, since you did 100 times here, it's 100 times 100, right? Which in the case of exponents, we can just add. 2 plus 2 is 4. 4 plus 1, because this is a 1 to 10 here, because this is now 1 into 9, right, is now 10 to the 5th. Another 1 to 10 dilution, 10 to the 6th, and then the last one ends up being a final dilution of 10 to the 7th, right? A tenth of a microliter is what we plate typically, right? So that's our volume plated in milliliters, right? These are possible counts. Which one would be countable? Y, right?
Oh, it's 30 or
30 or 300, right? 11's too few. What's our original culture, right? These are the things that we need. The number, right, of colonies divided by the volume plated multiplied by the dilution factor of that tube that we plated, right? So when you do out your math, you're allowed to use calculators. I'll give you guys relatively simple math, right? We get 1.26 times 10 to the 9 CFUs per milliliter. Units matter and scientific notation matters. we knew the initial volume, we could figure out the total potential CFUs in the original culture, which we didn't end up doing this semester. I never did get around to figuring how much media we started out with. So remember antibiotic sensitivity test, right? Go over that one. I'm not going to bore you guys with the details of that one right now. So we need to get to today's experiment. Yeah, there's a bunch of stuff in here. Okay. Ooh, I didn't even open today's. All right.